and I administer the Confocal machine that is supported through the RCMI program. And I'm just going to very, very briefly introduce you to the principles of confocal microscopy, what you can do with our machines. And then this afternoon, if any of you have further interest, you're welcome to swing by the Confocal lab. It's housed in the neurobiology core facilities on the second floor of the Adams building. And I'm happy to show you in more detail how it works and what we do. Confocal microscopy is a derivative of fluorescent microscopy. So while Dr. Wang was looking at whole animals, it's more for looking at the cellular level. And it visualizes fluorescent targets or fluorescent dyes, such as green fluorescent protein or fluorescently labeled antibodies. And as with all fluorescent molecules, they will absorb a photon of light and emit a photon of light. And there is always some energy loss in the process, so that the color of the light that is emitted is going to be of a lower energy or a longer wavelength than the color of light that was absorbed. And so when you're using fluorescent dyes, you find that each dye has a very specific excitation wavelength and a very specific emission wavelength. And so you can use them in combination and get different colored images based on the labels that you're using. We have two microscopes in the neurobiology core facility that allow fluorescent imaging. We have a conventional fluorescent microscope and a confocal microscope. Does this have a point on it?
You can still use your mouse.
And the concepts of how they work are quite different. The conventional microscopy, the excitation source is a white light, and the light is passed through a filter cube where all the colors except the excitation wavelength will be filtered out. And then there's a dichromatic mirror so that the excited light is sent down to your sample, and the fluorescent light then comes up through the mirror to the eye. So it's very, what you see is what you get. If it's emitting red photons, you see red photons, and your digital camera will image red photons. Confocal microscopy is quite different. The excitation sources are monochromatic lasers, so you need a different laser for each dye that you want to excite. And the acceptor for the fluorescent light, for the emitted light, is called a photomultiplier detector. And you have different detectors that will detect different colors. But the real thing that makes confocal, I can see it here. The real thing that sets confocal microscopy apart is that the emitted light is passed through a pinhole. And this pinhole eliminates all out-of-focus light. So what you see in this diagram is a specimen, and the light coming from this plane of the specimen goes through the objective and through the pinhole and reaches the detector. Light from these other planes would miss the pinhole and would not reach the detector. So that the image that is digitally displayed represents a very discrete optical plane of the sample as a whole. And so what you end up with, instead of individual images, are stacks of images that represent each different optical plane throughout the entire sample. So compared to conventional microscopy, where some cells will appear in focus and some cells will appear out of focus, you can take your stack of confocal images and collapse them into a single image, whereby you get optical resolution throughout the thickness of the entire sample. And this has great advantages when you are trying to discern cellular localization of proteins or co-localization of proteins. So here we have depicted two proteins. One is a nuclear protein and one is a cytoplasmic protein. And with conventional microscopy, you would not really be able to prove that this red nucleus is inside the green cell. It could actually be underneath. But with confocal microscopy, you can walk through the image planes one plane at a time, and if the two proteins fall in the same place within the same image plane, it's considered compelling evidence that they are in the same cell. And lastly, I just want to show that you can, because you have a stack of images that represents the entire Z thickness of the sample, you can then use software to reconstruct that entire three-dimensional sample. So here is a collapsed image of all the different planes, and we can isolate an individual cell and reconstruct it, and then use software to rotate it around. And in this example, we're trying to determine that that green fiber actually is coming into intimate contact with that cell. So again, I wanted to keep this very brief, but I'm happy to show any of you the instrumentation if you want to come by this afternoon, or of course you're all more than welcome to come use the machine. I'm happy to help you develop the methods for your projects and work with you to train you on the machine and work with some experimentation. Thank you.